Welcome back, students. We are going to start with the most important topic, and that was the last topic of chemotherapy, the anti-cancer drugs. So, the learning objectives for this topic include first, to study the pathogenesis of cancer, the genesis of cancer cells and their characteristics. We need to know this before actually jumping into the drugs. Second, to understand the general principles of cytotoxic anti-cancer drugs. And third, to study the pharmacology of anti-cancer cells. Okay, now let's start with the pathogenesis of cancer cells. Now, what is cancer? Now, cancer is a disease in which there is uncontrolled multiplication and spread within the body of abnormal forms of body's own cells. It is one of the major causes of death in the developed nations. There are more synonymous terms related to cancer like malignant neoplasm, cancer, or malignant tumor. They are all distinguished from the benign ones by the properties of tumors of dedifferentiation, invasiveness, and the ability to spread, that is, metastasize. There are three main approaches basically for the treatment of cancer. One is a surgical excision or irradiation and chemotherapy, of which chemotherapy is the main method for treatment and only few cancers, but it is increasingly used as an adjunct to surgery or irradiation for many types of tumors. Now, other approaches in the cancer treatment are like immunotherapy or by use of angiogenesis inhibitors, we'll come to that, or gene therapy, or by using certain biological response modifiers which will boost your immune system. So these are some other approaches, but the main approaches is by surgical excision or irradiation or chemotherapy, of which chemotherapy is the first approach for the treatment of cancer. Now let's look at the special characteristics of a cancer cell. Now this is very interesting. Now there are four main features which distinguish from a normal cell and a cancer cell. One is uncontrolled proliferation. Second is dedifferentiation or loss of function. Third is invasiveness. And fourth is the ability to metastasize. Now first, let's look at the first one, the uncontrolled proliferation. Now the proliferation of cancer cells is not controlled by the processes that normally regulate the cell division and the tissue growth. It is rather than their rate of proliferation that distinguishes from the normal cells. See, even normal cells, they proliferate, they undergo the cell cycle, but it is the only difference between the cancer cell and the normal cell is that the rate of proliferation, how fast do they proliferate, how fast do they multiply. For example, some normal cells such as neurons have little or no capacity to divide and proliferate. But others, for example, in the bone marrow and the epithelium of the gastrointestinal tract, have the property of continuous rapid division. Some cancer cells multiply slowly, like those of the plasma cell tumors, and some of them they multiply very fast, like the lymphoma cells or the blood cancer. Now, let us come to the cell cycle. Now, this diagram represents cell cycle, specifying four phases: the G1 phase, the S phase, the G2 phase, and the M phase, indicating the main sites of action of the cell cycle. Now, in a normal cell, a cell is stimulated to divide by certain growth factors, which starts in the G1 phase and prepares for DNA synthesis. Actually, this entire process is regulated by two forces. Entire cycle is regulated by two forces. One is called as a positive regulatory forces, wherein because of this only, there is initiation of cell division and multiplication. Now, these positive regulatory forces are nothing but the growth factors or cyclins and cyclin dependent kinases, CDKs. So, these are the ones which stimulate the process of cell division. Whereas on the other side, it is well balanced by negative regulatory forces also. Now, these regulatory forces, they act on certain checkpoints, wherein these forces, once they get the signal that something is wrong while in the process of cell division, they will get activated. These are nothing but certain genes like retinoblastoma gene action, RB gene that is called, or P53 gene action. They act as policemen, at checkpoints. So you have a cell stimulated to divide by the growth factors, which starts at the G1 phase, and this is carried forward to the synthetic phase by the CDKs, cyclin dependent kinases. And at some point, if it is felt that the cell is not multiplying properly, then the negative forces will be involved in the cell cycle process, and proteins are induced by various genes like the P53 gene and the retinoblastoma gene. And if there is a DNA damage, as we said, the actions of the tumor suppressor gene, that is a P53, will stop the cycle at checkpoint and allow the cells to go for a repair. But if this repair fails, then apoptosis, which is nothing but it is a inbuilt process of cell destruction of self-destruction mechanism of the cell. It is a programmed series of event. Apoptosis, you must be aware of. Apoptosis is inbuilt process of self-destruction mechanism of the cell consisting of genetically programmed sequence of biochemical events leading to cell death. So this is initiated. Now what is this G0 phase? G0 phase represents a phase in which the cells are not dividing, but they can re-enter the cycle. Now, now, the uncontrolled proliferation of the cancer cell, we need to consider the signal transduction mechanisms for cell division as well. Now, the cells in multicellular organisms divide in response to the extracellular signals, that is the growth factors, or the cell surface receptors, which can trigger the intracellular transduction events involving both the transducers and finally the production of cell cycle transducers, which lead to DNA synthesis and finally to cell division. Now, one reason for continuous proliferation of cancer cells is a property called as immortalization. This is thought to be in contrast to normal cells, their telomeres do not shorten with each round of cell division. So in normal cells, the telomeres, they shorten at each round of cell division. And this does not happen in a cancer cell. The shortening is possibly disappearance of telomeres is believed to be the basis of cessation of cell division. So this shortening is responsible for abrupt cessation. Cessation means closure of cell division, leading to complete form of cell. But in a growing tissue, an enzyme called as telomerase maintains and stabilizes the telomeres. But most fully differentiated cells do not express this telomerase enzyme at all. But about 95% of late stage malignant tumors, they express this telomerase. And it is suggested that this enzyme is responsible for producing immortality. Matlab they never die. They keep on multiplying, multiplying, and multiplying on the cancer cells. So the involvement of telomerase enzyme is equally important. Telomerase levels in cancer cells, they are abruptly more. One more interesting point to be noted with cancer cells is that is a process of angiogenesis. Now, what happens is the actual growth of the tumor depends upon the development of its own blood supply. So, tumors of 1 to 2 millimeter in diameter roughly can receive nutrients by diffusion, but any further expansion of the tumor will definitely require development of new blood vessels called as angiogenesis. Now, this process of development of new blood vessels only to cater to the needs of the cancerous tissue is called as angiogenesis. This occurs in response to vascular growth factors which are produced by the growing tumor. And during angiogenesis, there are certain enzymes called as metalloproteinases which break down the surrounding tissue, making space for the new blood vessels. So, very interesting facts what we have seen in controlled proliferation, the cell cycle. Then we have seen about what are the growth factors which are involved in uncontrolled proliferation. Then we have talked about the telomeres, about angiogenesis and metalloproteinases. Now, looking at the second property of cancer cell, loss of function, that is dedifferentiation. What happens is one of the main characteristic of the cancer cell is the loss of differentiate of different tumors. The capacity to differentiate is lost. See what happens is when the cells are normally dividing from the stem cells, as time moves on, these cells differentiate into various levels of relevant tissue. Now, for example, they get programmed a series of functions getting differentiated into the cells of the heart, the cells of the hands, the cells of the legs, and all. Now, this property is lost by the cancer cells. Next is invasiveness. Now, normally, you will not find heart cells in liver or liver cells in some other gastrointestinal tract region. Now, normal cells are not found outside their designated tissue of origin. For example, that's what I said. Liver cells are not found in bladder, or pancreatic cells are not found in testis. This is because during differentiation and during the growth of tissues and organs, normal cells develop certain spatial relationships. Spatial means area. They have respect for each other's boundary, and therefore these relationships are maintained at various tissue specific survival factors. Once they leave the boundary means they are subjected to apoptosis, so they do not leave their boundary and go. And this property is not carried by the cancer cells. So because of the involvement of metalloproteinases also, the cancer cells they rapidly spread to the other organs as well, and they keep on invading the other nearby tissues. Next property is metastasis. What is metastasis? Now, these are secondary tumors which are formed by the cells that have been released from the initial or the primary tumor. So, if the cancer from the stomach reaches the gastrointestinal tract, that forms the secondary tumor. So, the primary tumor is from the stomach. So these are secondary tumors which are formed by the cells and they have been released from the initial of the primary tumor and they have reached the other sites through the blood vessels or lymphatics as a result of being shed into the body cavities. So you have these four characteristics of this. So basically the genesis of a cancer cell, um, two main categories are involved for this genetic change. One is the inactivation of the tumor suppressor gene and activation of the proto-oncogenes to oncogenes. Oncogenes are cancerous genes. These genes they get converted into the actual cancer genes. They are called as proto-oncogenes getting converted to oncogenes. So the next diagram in the next slide will give you a clear picture about this explanation. Okay, how cancer is being formed. Let's see that. So in this diagram, we see that how the signal transduction pathway or the genesis of cancer is involved. You can have dedifferentiation also occurs in this. The cell proliferation signals here they include the growth factors acting on the receptors and the subsequent action on the cytosolic, nuclear, and cell cycle transducers. In this, the genesis of cancer is usually multifactorial. You have chemical viruses or irradiation, which stimulates the acquired mutations to alter the gene expression. This can also be done by the inherited mutations. Now, this altered gene expression leads to conversion of proto-oncogenes to oncogenes or a decreased expression of tumor suppressor genes. And this, along with the other intracellular factors, either causes alteration of a normal cell proliferation signal, or the apoptotic signals may not be responsive. These are the intracellular effects which are taking place, leading to loss of cell cycle control. This will give rise to uncontrolled cell proliferation, the tumor development, and production of vascular growth factors, and the involvement of metalloproteinases, angiogenesis, telomerase enzymes, etc., leading to tumor growth, invasion, metastasis, finally leading to a condition called as cancer. So this diagram explains about the mechanism where the anti-cancer cells act. See, most of the anti-cancer drugs act mainly during the DNA synthesis of the cell cycle. That is the synthetic phase, that is the S phase. Alkylating agents or antimetabolites or cytotoxic antibiotics, they act in the synthetic phase. So synthetic phase here is comprised of the synthesis of purine, pyrimidine getting converted to ribonucleotides, deoxyribonucleotides, DNA to RNA, RNA to proteins, proteins getting converted to enzymes and microtubules. So there are various points or uh, wherein the anti-cancer drugs can act. This diagram, this picture, this slide represents where these drugs actually involving which enzymes they are involved in the process of inhibiting the. Now coming to the classification of drugs used in the treatment of cancer chemotherapy, they are generally classified into 3 to 4 categories. The first main category is the cytotoxic drugs, which include alkylating agents, antimetabolites, cytotoxic antibiotics, and plant derivatives. Second category is the hormones, and third category is the miscellaneous agents involving some other enzymes and effects. So let's come to the first category of cytotoxic drugs, the alkylating agents and related compounds. Alkylating agents and these compounds, related compounds, contain basically a chemical group which have the property of forming covalent bond with suitable nucleophilic substances in the cell. Now, with alkylating agents themselves, the main step is the formation of carbonium ion, which is nothing but a carbon atom with 6 electrons in the outer shell. Now, such ions are highly reactive and they react immediately with an electron donor, such as an amine or an OH group or an SH group. Now, most of the cytotoxic anticancer alkylating agents are bifunctional, that is, they have 2 alkylating groups. The N7 nitrogen of guanine is strongly nucleophilic and is the main molecular target for alkylation in DNA. You can see the diagram. So cross-linking can cause. Now what happens is with 2 nucleophilic sites, cross-linking takes place A bifunctional alkylating agent causes intra cross-linking, and this cross-linking can cause defective replication due to pairing of alkyl guanine with thymine, leading to the substitution of AT for GC. And finally, the chain breaks. So therefore, there is no DNA synthesis. And this effect, all these effects of alkylating agents, they principally occur during the DNA synthesis. The unwanted effects of these alkylating drugs include mainly myelosuppression, sterility, and the risk of leukemia. The main alkylating agents are nitrogen mustards. Example is cyclophosphamide, which in vivo gets activated to aldophosphamide, which is then converted to phosphoramide mustard, which is a cytotoxic molecule, and acrolein, which causes bladder damage, and this can be actually taken care by another drug called as mesla. Now, cyclophosphamide myelosuppression affects particularly the lymphocytes. Another drug coming under this alkylating agent is nitrosoureas, lomustine, carmustine type of drugs. They act on non-dividing cells. They can cross the blood-brain barrier and cause myelotoxicity also. Another group of compounds from the platinum compounds is this is platin, which causes intra-strand linking in DNA. It has got a low myelotoxic effect and causes severe nausea and vomiting and can be nephrotoxic. This has revolutionized the treatment of germ cell tumors. So under nitrogen mustard, so under alkylating agents, you've got nitrogen mustard, cyclophosphamide, aldophosphamide type of drugs, nitrosoureas, and the cisplatin category of drugs. Well, coming to the next class of cytotoxic drug, the antimetabolites. Uh, Under this, antimetabolites are basically drugs which will block the pathways or they will divert the pathways for DNA synthesis. Now, the first category are the folate antagonists. Now, the main folate antagonist is methotrexate. It is one of the most widely used antimetabolites in cancer chemotherapy. Now, we are aware that folates are essential for the synthesis of purine nucleotides and thymidylate, which in turn are essential for DNA synthesis and cell division. Now, in this diagram, you can see that the tetrahydrofolate polyglutamate functions as a carrier of a one carbon unit, providing the methyl group the necessary for conversion of DUMP to DTMP, that is 2-deoxyuridylate to 2-deoxythymidylate by thymidylate synthetase. And in this process, the one carbon transfer also results in oxidation of tetrahydrofolate to dihydrofolate. So you can see the important role dihydrofolate reductase enzyme and the thymidylate synthetase enzyme plays in order to produce the um, essential metabolite required for synthesis of DNA and cell division, further cell division. Now, methotrexate is a drug which has got a very higher affinity for dihydrofolate reductase than for dihydrofolate. So it thus inhibits the enzyme and depletes the intracellular tetrahydrofolate. So the binding of methotrexate to DHFR actually involves an additional hydrogen bond or ionic bond, which is present when the dihydrofolate binds. This reaction is most sensitive to tetrahydrofolate depletion, which is thymidylate synthesis, and is inhibited at a methotrexate concentration of as slow as 1 micromole per liter. Now, what's happening is the unwanted effects of this are myelosuppression and possibly nephrotoxicity with these drugs. So, methotrexate inhibits dihydrofolate reductase, preventing generation of tetrahydrofolate. This is the main result in interference with thymidylate synthesis. Another class of drugs like pyrimidine analogs, like fluorouracil, is converted to fraudulent nucleotide and inhibits thymidylate synthesis. Cytarabine, its triphosphate form, inhibits DNA polymerase. It is a potent myelosuppressive. Coming to the purine analogs, mercaptopurine is converted into fraudulent nucleotide. Fraudulent means false nucleotide. Fludarabine in its triphosphate form inhibits DNA polymerase. It is a myelosuppressive. And the drug pentostatin, also inhibits adenosine deaminase, which is a critical pathway in purine metabolism. Well, the next class of drugs are cytotoxic antibiotics. Um, the main anticancer anthracycline antibiotic, that is a cytotoxic antibiotic, is doxorubicin. Doxorubicin has several cytotoxic action. It binds to the DNA and inhibits both DNA and RNA synthesis, but its main action appears to be mediated through an effect on topoisomerase 2, which is also called as DNA gyrase. The activity of which is increased in the proliferating cells. The significance of this enzyme actually lies in the fact that during replication of the DNA helix, normally there is a reversible swiveling movement which need to take place around the replication fork in order to prevent the daughter DNA molecule becoming entangled during mitotic segregation. And this swiveling is produced by topoisomerase 2, which cuts both the DNA strands and subsequently reseals the breaks. So doxorubicin intercalates in the DNA and its effect is to stabilize the DNA topoisomerase 2 complex or DNA gyrase enzyme after the strands have been nicked. So this will prevent the further steps involved in DNA synthesis. Bleomycins. Now, just before going to bleomycins, the main unwanted effect of doxorubicin is that it leads to dose-related cardiac damage. This is very peculiar, leading to dysarrhythmia and heart failure. This action is due to generation of free radicals. The bleomycins are a group of metal-chelating glycopeptide antibiotics that degrade preformed DNA, causing chain fragmentation and release of free bases. Their action on DNA is thought to involve chelation of ferrous ion and intercalation with oxygen, resulting in the oxidation of iron and generation of superoxide or hydroxyl radicals. It is most effective in the G2 phase of the cell cycle and mitosis, and it is also found to be active against non-dividing cells in the G0 phase. The unwanted effect of bleomycin is its most serious toxic effect, that is pulmonary fibrosis, which occurs in 10% of patients treated with this drug. The other drug, dactinomycin. Dactinomycin intercalates in the minor groove of DNA, which is between adjacent guanosine cytosine pairs, interfering with the movement of RNA polymerase along the gene and thus preventing transcription. There is also evidence that it has a similar action to anthracyclines on topoisomerase 2. And the unwanted effects of this drug include nausea, vomiting, and myelosuppression. Mitomycin, another drug which after enzymatic activation in the cells functions as a bifunctional alkylating agent acting at the O6 position of guanine. So it cross-links DNA and also degrades DNA through the generation of free radicals. This drug also causes marked myelosuppression and can cause kidney damage and fibrosis of the lung tissue. Well, coming to the next class of anti-cancer drugs, the plant derivatives and hormones. Amongst the plant derivatives, vinca alkaloids are famous as anti-cancer drugs. Now, vinca alkaloids are derived from the plant called as periwinkle plant. We are aware of this. And the main alkaloids are vincristine, vinblastine, and vindersine. They act by binding to the tubulin and inhibit its polymerization into microtubules, preventing spindle formation in mitosing cells and causing arrest at the metaphase. So these drugs they act at the not in the synthetic phase but at the metaphase of the cell cycle by involving tubulin and inhibiting the polymerization of these microtubules, which are an essential stage in the mitotic phase of cell division. Now these effects are very uh, obviously seen during the mitotic phase, and they also inhibit other cellular activities that involve the microtubules, such as the leukocyte phagocytosis and chemotaxis, as well as axonal transport in the neurons. Um, etoposide is another alkaloid which is derived from mandrake root. It acts by inhibiting the mitochondrial function and of nucleoside transport, as well as an effect on topoisomerase 2, similar to that of doxorubicin. Then we have drugs called like paclitaxel, which are derivatives of yew tree bark. They act on microtubules, again stabilizing them in the polymerized state. Unwanted effects include bone marrow suppression and neurotoxicity. Drugs like irinotecan, which belong to camptothecins, bind and inhibit topoisomerase 1, which occur at high levels throughout the cell cycle. Now, talking about hormones, now hormones, tumors derived from hormone sensitive tissues will definitely be hormone dependent. Like, for example, the breast tissue or the tumors of the pineal gland or the testes, they will all be hormone dependent. So their growth can be inhibited by hormones with opposing actions. Now, these hormone antagonists inhibit the synthesis of relevant hormones. So, hormones or hormone analogs, which themselves have inhibitory action on particular tissues, can be used in the treatment of tumors of these tissues. For example, glucocorticoids. They have inhibitory effect on lymphocyte proliferation. So they are used in leukemias and lymphomas. Or tamoxifen. Now this drug is used for breast tumors because in case of breast tumors, estrogen is the main hormone which will favor estrogen dependent breast tumors. So tamoxifen will have the opposite action of estrogen. Or gonadotrophic releasing hormones analogs for prostate and breast tumors are used. Anti-androgens are used for prostate cancers. Or inhibitors of sex hormones synthesis for postmenopausal breast cancers. So these are some of the examples wherein hormones also play a very important role in controlling the cancer of those tissues which are hormone dependent. Now, apart from plant derivatives and hormones, we can also have radioactive isotopes. They can also be targeted at specific tissues like iodine 131 for thyroid tumors. This is very safely used in treating thyroid tumors. There is one more class under anti-cancer drugs, the monoclonal antibodies and protein kinase inhibitors. This is a recent one. Many tumors overexpress growth factor receptors. Now, tumors they overexpress growth factor receptors, and therefore they stimulate definitely the cell proliferation and tumor growth. So this can be inhibited by monoclonal antibodies, which bind to the extracellular domain of these growth factors, the oncogenic receptor HER2, or which neutralize the growth factor themselves. Another group of drugs like protein kinase inhibitors, they are used in they basically involve the downstream signaling which are triggered by the growth factors. So extracellular part, when the growth factor is triggered by certain agents, will initiate certain events intracellularly. This is taken care by the protein kinase inhibitors. They will inhibit specific oncogene kinases or specific receptor tyrosine kinases or other receptor associated kinases which are involved in signal transduction mechanisms intracellularly. And some monoclonals, they act also on lymphocyte cell surface proteins to cause lysis, thereby preventing proliferation. So we have seen about anti-cancer drugs. The most important ones are alkylating agents, anti-cancer antibiotics, the plant derivatives and hormones also we have seen, and the newer ones like monoclonal antibodies, protein kinase inhibitors, and also biological response modifiers like interferons are also being used in the treatment of cancer. Okay, I hope you'll have enjoyed this very important topic on anti-cancer drug. It must have been slightly lengthy, but I wanted to cover that because this has already been taken care in pharmacochemistry as well. So this would have been nothing but another revision in pharmacology as well. Okay, thank you.